GMAN is going to be starting his science series today, and the first topic is going to be abiogenesis. Now, knowing GMAN, he'll make reference to abiogenesis in the beginning for maybe about 2 minutes, and then the whole thing's going to turn into evolution. And it will be the same old thing. How do you get new anatomical features from genetic mutations? If you believe in evolution, you believe you're related to a potato, okay, and all that nonsense. Now, as far as the new anatomical features from genetic mutation, G-Man's been asking that question for years. And I remember the first time I'd ever heard G-Man ask that question. I remember my reaction was, what? Like, something didn't sound right there. I can't tell you what it is, but something didn't sound right. And, well, there's a reason for it. Okay, well, G-Man, the new anatomical features from genetic mutations. I know I'm putting you on the spot here. Do you actually remember where you learned that? Like, was it in school, or is it something you learned later on your own?
I learned that on my own.
You learned that on your own. Okay.
Yeah.
So that was G-Man's best attempt at formulating his own question. And still the reaction from it is, what? But G-Man will be asking, how do you get new anatomical features from genetic mutation? He will be asking to have people come in to convince him. He will say, I can be proven wrong. He'll say, I want to learn. All of that is an absolute lie. Okay? All of you already know, people have explained this stuff to him over and over again. But on his very show, he actually had someone qualified to do it, and he actually explained it. And just as you would expect, in one ear, out the other. In fact, not even that. I've never even heard him speak of that man again. Because he gave G-Man what he wanted. He explained it to him. I'll show you.
So exactly, how do you get new anatomical features from mutations? Before you answer the question, do me a favor and tell everybody what a mutation is, how mutations work, and then explain your explanation on how you get new anatomical features.
You got it. So that is such a fun question. So first thing, what a mutation is. The short version is a mutation is just, and by the way, I'm going to interrupt myself to just say, after I give this answer, I want to give other people a chance to talk and ask questions because I don't want to monopolize this conversation, but I just want to let me just give a kind of, so a mutation is any heritable change to the sequence of DNA in an organism. And that can be just an A changes to a G within the sequence of nucleotides. It could be a gene duplication. It could be like a chromosome, like a structural change, like a translocation. Any change to the sequence of the DNA, that is a mutation. There's small scale, there's large scale mutations. What was the second question there? Sorry, it was what is a mutation? What does it do?
How do you get new anatomical features?
How do you get new stuff? So the way you get new stuff is really cool. And this gets into a field called EVO-DEVO, evolutionary developmental biology. And the really cool thing is that there isn't it's a common misconception that there's a gene to make eyes, there's a gene to make legs, there's a gene to do X, there's a gene to do Y. And what we actually see is that there are genes that kind of set patterns. So there's a gene that says extend away from the midline of the body. There's a gene that'll say fold inwards. And the kind of the timing and the location of the expression of all these different genes in combinations gets you the different structures. And so, for example, I'll use the eye as an example, because this is the nice thing about eyes is that you can look at all the different organisms that have the ability to like detect light in some capacity, and you could see basically the evolutionary history of eyes across those different organisms. So there's a group of proteins. It doesn't matter how they work. They're called opsins. They detect light. That's all that matters. And you can express those genes. They're evolved from another set of proteins. It doesn't matter what the other set does. It doesn't matter. And basically, you could picture a flat surface, right? And you detect light. Okay, like you could detect light, dark. That's fine. But what if you had, instead of a flat surface, what if you had the signal that says fold inwards? Well, folding inwards is a very common signal developmentally, because that's a way to increase surface area. And increasing surface area is like, we like that. That's great for absorption or exchange. So like gills, intestines, you want to fold in. So let's say you fold in on the surface. Now you have your opsins in, a cup. Well, now that's useful because now you can detect the direction of the light, right? Is it coming from this direction or is it coming from this direction? That's useful. Now, along with those opsins and the folding pattern, you can have a layer on top. It's easy to have two layers of cells, and for one to say fold in and the other one to not fold in. Well, now you can have a translucent layer on top and a folded in layer on the bottom. This is starting to sound like an eye. And you can go through these different stages without needing any specific like new genes, but you're just changing the regulatory pattern of how they're expressed and where and when during embryonic development, and you get a much more complex structure. And you can look at this from top to bottom. So you could look, for example, at the development of limbs as the same kind of thing, where you're looking at like extend from the body. Okay, well, the simplest things that extend from the body have like little nubs, right? Think about like a caterpillar leg. It doesn't have like true legs. It's just like little nubs, right? But then if you can look at other groups, arthropods, mature arthropods, insects, spiders, that kind of thing, well, they have true legs. Mammals, true legs. Reptiles, true legs. Well, the really cool thing is they have the same genes that say build a leg here, but it's not a gene that says build a leg here. All that gene says is extend here. And if you express that gene for a long enough time, it causes the tissue to split and grow in two directions. So that's how you get like digits, for example, in your fingers. So our arms aren't just one continuous thing. It's at some point it splits into multiple things that grow away from the body. So and I apologize, this is not a short answer, but the short-ish answer to how do you get new information is you have a pre-existing thing, and we could talk about where that comes from because there have been lots of experiments on that, but you have a pre-existing thing that does a thing, extend from the body, build X, build Y, and you change the pattern of where and when that's expressed within a multicellular organism, and that results in a new feature in that organism.
And for how long, right? It's where, when, and how long is it expressing.
But it's